Today we will discuss the histology of respiratory system. Within respiratory system, we generally include the histology of trachea and lungs. And in the lung section, you will see the sections of alveoli and different type of bronchioles. Okay, terminal bronchioles and other bronchioles you will see, and some part of the bronchus you can also see in the section of the lung. Okay, so all those we will discuss today. So we will discuss the histology of lung, trachea, bronchi, terminal bronchiole, respiratory bronchiole and the alveoli. Okay. So the trachea is a cartilaginous tube that acts as air passage from larynx to the lung. Length is about 10 centimeter, diameter is about 2.5 centimeter outer diameter and 1.5 to 2 centimeter inner diameter. Okay. And the trachea divides into two main primary bronchi or two principal bronchi, and the lumen of trachea is non-collapsible, that it remains open due to the presence of the cartilages. And the unique feature of trachea is presence of C-sepate hyaline cartilages. Okay, that's how you differentiate trachea from other bronchus and bronchioles. Presence of this cartilage is important for trachea. Any queries? Okay, so what you will see in the wall of the trachea, there is mucosa from inside to outward we are discussing. First you will get the mucosa, then submucosa, then cartilage and muscular layer, and there you will get the adventitia, outermost layer. Okay, adventitia is loose connective tissue layer. So first we will discuss the mucosa. Trachea is lined by ciliated pseudostratified columnar epithelium. Very important. This is identification feature of the trachea. Okay, and this is very frequently asked in your Viva and MCQ. This lining epithelium, pseudostratified ciliated columnar. And what are the cells of tracheal epithelium? Tracheal epithelium shows these following cells: ciliated columnar cells, mucus cells, brush cells, small granule cells and basal cells. All these are present in the tracheal epithelium. Next, underneath this epithelium there will be basement membrane. The thick basement membrane is a characteristic feature of trachea. In hematoxylin eosin staining, the basement membrane is present as a glassy or homogeneous eosinophilic pink layer. Okay. Lamina propria, it consists of loose connective tissue. It may show diffuse lymphocytes and some lymphatic follicles in the form of bronchus associated lymphoid tissue, BALT. Okay. In case of gut, there is gut associated lymphatic tissue, that is known as GALT. Okay. So like this, some lymphatic tissues are present in different systems. And the lamina propria also contains numerous elastic fibers. So we have completed the mucosa. After that you get the submucosa. On HE staining, it is difficult to distinguish the demarcation between lamina propria and submucosa. No proper demarcation line between these two, because both have containing loose connective tissue, and submucosa contains mucus acini with serous demilunes, and duct of these glands are lined by cuboidal epithelium. Okay. Submucosa also contains lymphatic follicles, blood vessels and lymphatics. Okay. Tracheal cartilage and trachealis muscle. Trachea contain about 16 to 20 C-sepate hyaline cartilage rings. These numbers is also sometime asked in your Viva. How many cartilaginous rings are present in trachea? Okay, so the gap of C-sepate cartilage, it is bridged by fibroelastic tissue and trachealis muscle, that is a smooth muscle. With advancing age, hyaline cartilage may get calcified. Hyaline cartilage consists of chondrocytes lying in lacuna surrounding the homogeneous matrix and perichondrium. Outer fibrous layer, perichondrium has two layers, we have learned during the cartilage class. It has outer fibrous layer and inner cellular layer. Next we go to the adventitia, outermost layer. It is fibroelastic connective tissue layer that contains the blood vessels and nerves. Okay, understood? So these are the 4 layers you will get in trachea. Mucosa, lining epithelium is important, submucosa, then trachealis, cartilage and muscle layer, and then you will get the adventitia. Okay, so these are the 4 layers of trachea. This is a schematic diagram of trachea. From inside, this is the lining epithelium, pseudostratified ciliated columnar epithelium. Below it, here is the lamina propria, then there is submucosa, then here is the C-shaped cartilage, the pink one is the cartilage, and the 2 ends of the cartilage is bridged by fibroelastic tissue and trachealis muscle. Okay, and these are the sections of some blood vessels, and here outermost is the adventitia, the green lines you can see, these are the adventitia. These are the 4 layers from inside outward. See, this is the lining epithelium, pseudostratified ciliated columnar. You can see the cilia and the nuclei in different layers, but all the cells are reaching the basement membrane, thick basement membrane you can see here. And these are the goblet cells or mucous cells are also present here. This is also a presenting feature of this epithelium. Like this you will see under the microscope. Okay. So this is lining epithelium, pseudostratified ciliated columnar epithelium, then lamina propria, then submucosa. In the submucosa you can see this seromucous gland. Okay. Mucus acini with serous demilunes. The deep colored ones are the serous demilunes and light colored ones are the mucus acini. And this is cartilage, a part of cartilage has been shown. You can see the chondrocytes, the matrix, okay, and the perichondrium having 2 layers. And this is trachealis muscle, spindle shaped smooth muscles you can see here. And outermost is the adventitia containing loose connective tissue and some adipose tissue here. Okay, understood? So these are the layers you will see in the wall of the trachea. So this is summarization of histology of trachea. You will see the mucosa. Mucosa is having lining epithelium and lamina propria. Lining epithelium is pseudostratified ciliated columnar epithelium with goblet cells, brush cells, small granule cells and basal cells. Lamina propria contains bronchus associated lymphoid tissue, vault, okay, separated from the submucosa by elastic membrane. And submucosa contain connective tissue and mucous glands. There will be 3rd layer is cartilage and muscle, C-shaped hyaline cartilage and trachealis muscle. This is asked which type of cartilage is present in trachea. This is hyaline cartilage, okay. And trachealis muscle is smooth muscle. This is present in the 3rd layer. And there is adventitia. Adventitia contain fibroelastic tissue. The basement membrane of trachea is very thick and there is bronchus associated lymphatic tissue. Okay. So like this you will see under microscope. This is the lining epithelium, pseudostratified ciliated columnar epithelium with goblet cells, then lamina propria, submucosa, within the submucosa some glands you can see. Then you will see the cartilage, this homogeneous layer with dots of the chondrocytes. These are the nucleus of the chondrocytes you will see, and adventitia containing section of blood vessels, connective tissue, nerves, like this. So like this you will see under microscope. Next after the trachea we go to the bronchial tree. Bronchial tree you have learnt that chart. So trachea divides into 2 principal bronchi or primary or main bronchi, right and left. Right bronchus is wider and shorter than the left bronchus. Right bronchus divides into 3 secondary or lower bronchi, and whereas left bronchus into 2 secondary bronchi. Secondary bronchi supply the lobes of the lungs, 3 lobes in right lung, 2 lobes in the left lung. And the secondary bronchi divide into 10 segmental or tertiary bronchi that supply the bronchopulmonary segments of the lungs. Tertiary bronchus divides into numerous bronchioles. The smallest bronchioles are called the terminal bronchioles, and terminal bronchioles divide to give rise to respiratory bronchiole and finally alveolar duct and alveoli. Up to the terminal bronchiole, as there is no gaseous sections, this part of the respiratory tract is called the conducting part. Okay, up to the terminal bronchiole, conducting part. and the terminal bronchiole and distal to it, that occurs where occurs the gaseous section. Hence, this part is known as respiratory part. Okay. See the division: trachea, two principal bronchi, secondary bronchi or lobar bronchi, segmental or tertiary bronchi, multiple divisions up to terminal bronchiole, and after that bronchioles are having no cartilage, and up to terminal bronchiole it is conducting part, rest of the part is respiratory part. Okay. Okay, so this chart you have seen previously. Trachea, two principal primary bronchi, secondary or lobar bronchi, 3 on right, 2 on left. Tertiary or segmental bronchi, 10 on each side, numerous bronchioles are present. And then there is terminal bronchiole, respiratory bronchiole, alveolar duct and alveoli. Green part is respiratory part, gaseous exchange occurs here, and the red colored one is conducting part, no gaseous exchange. This is the respiratory part. Okay. Terminal bronchiole, respiratory bronchiole, alveolar duct, alveoli. Okay, alveolar sacs. Looks like a bunch of grapes. Okay. Next we go to the lung. Lung are a pair of organs that are principal site of gaseous exchange, and it consists of pleural covering, connective tissue septa and lung parenchyma with airway passage. Principal bronchus divides into lobar bronchus that enter the lungs to form the intrapulmonary bronchi, and the lung parenchyma consists of bronchioles and alveoli. Intrapulmonary bronchi include the secondary or lobar bronchi and tertiary or segmental bronchi. Segmental bronchi divide into many small bronchi that finally form the bronchiole. Bronchiole diameter is less than 1 millimeter and no cartilage in the wall. Histology of intrapulmonary bronchi. It has these following layers: mucosa, muscular layer, submucosa, cartilage and adventitia. Okay. This muscular layer is added here. That is the difference from the trachea. Okay, understood? So, intrapulmonary bronchi, mucosa consists of pseudostratified ciliated columnar epithelium with goblet cells. In respiratory system, when it is getting more deeper towards the alveoli, there is change in epithelium. Okay. Up to intrapulmonary bronchi, there is pseudostratified ciliated columnar epithelium. After that, you will get only ciliated columnar epithelium. Then you will get ciliated cuboidal epithelium, then only cuboidal epithelium, then in alveoli squamous epithelium. Okay, like this there is gradual changing of the epithelium from conducting part to the respiratory part, and this change is due to their different functions. Okay, in conducting part you need conduction of air and removal of the foreign bodies and mucus from the respiratory tract, and in the respiratory part you mainly need the gaseous exchange more. So, there is cuboidal to squamous epithelium is present in the areas where gaseous exchange is occurring. Okay, understood? Okay, so we will discussing the intrapulmonary bronchi. It consists of pseudostratified ciliated columnar epithelium with goblet cells. Height of the epithelium decreases as the size of the bronchus decreases. Lamina propria contains many elastic fibers and seromucous glands. Muscular layer, it is a smooth muscle layer and forms a complete circumferential layer around the epithelium in the intrapulmonary bronchi. Smooth muscle fibers are arranged spirally and help to maintain proper diameter of the airway passage. Next we go to the submucosa. It consists of loose connective tissue with seromucous glands. Then cartilage layer, intrapulmonary bronchiole shows the presence of discontinuous cartilage plates. Here C-shaped cartilage is not present. Cartilage plates are not continuous. There is breaking in between. Okay. So, the cartilaginous plates help to keep the bronchi open. Adventitia, cartilaginous plates are externally surrounded by a connective tissue layer containing blood vessels and lung parenchyma, that is known as adventitia. So, these are the 5 layers of intrapulmonary bronchi. This muscle layer is extra added from the trachea. So, this is summarization of intrapulmonary bronchi. mucosa, pseudostratified ciliated columnar epithelium with goblet cells, then lamina propria. Epithelium and lamina propria together forming the mucosa. After that, you get the smooth muscle layer, spirally arranged fibers are present. Then some mucosa, loose connective tissue layer with seromucous glands, then cartilage layer, it is discontinuous plates, then you get the adventitia outermost. Okay. This is a section of intrapulmonary bronchus. This is epithelium, below the epithelium lamina propria, then these are the muscular layer containing smooth muscle fibers, spindle shaped fibers you are seeing. Then submucosa, you get the seromucous glands. These are the cartilage plates in the cartilage layer, and after that you get the adventitia, outermost sections of blood vessels. Okay, understood? And this is a terminal bronchiole. Here you get no cartilage. In bronchiole there is no cartilage. You have learned. So you get the mucosa, then lamina propria, then muscle layer, then submucosa, and then the adventitia. Okay, outermost or connective tissue layer. So bronchioles are the airway tubules that are less than 1 millimeter in diameter, and their wall does not have any cartilage. That is the difference from bronchus. Okay, that it does not have any cartilage in bronchiole. And some mucosal glands. Pulmonary lobule bronchiole further divide to give rise to the terminal bronchioles. Pulmonary acinus terminal bronchiole further divides into respiratory bronchiole and alveoli. Respiratory bronchoalveolar unit, it is the functional unit of lung and consists of a single respiratory bronchiole and the alveoli supplied by it. Okay, these together forming the respiratory bronchiolar unit. These are the divisions are showing segmental bronchus from their larger bronchiole. It is dividing multiple times up to the terminal bronchiole, then respiratory bronchiole, then alveoli. Okay. So what is the structure of bronchiole? Bronchiole consists of these following layers: mucosa, smooth muscle layer, and connective tissue layer. There is no cartilage, very important, no cartilage. How you will identify? Okay, if special slide is given from lung, this can come as a OSPI question, you have to write, or in theory if it comes as a short note, histology of lung, you have to write down these points. Okay. So mucosa, larger bronchiole is lined by ciliated. Here the changes is showing. Okay, changes of epithelium is written. Larger bronchioles, it is lined by ciliated pseudostratified columnar epithelium with goblet cells. Medium bronchiole, simple ciliated columnar epithelium with few goblet cells. Smaller or terminal bronchiole, simple cuboidal epithelium with Clara cells and without goblet cells. Okay, so these are the lining epithelium of different bronchioles. Okay, so what is absent in bronchioles? Bronchioles does not have cartilage in their wall, and bronchioles do not have seromucous glands in lamina propria, and terminal bronchioles do not have goblet cells. Okay, so these are important for your viva and MCQs. Next is Clara cell. Clara cell sometimes come as a short note. Okay, it is present, it is asked in your viva. Okay, Clara cells. In lung viscera also it can be asked. Lining epithelium, different part of the bronchiole like this, or Clara cells, their functions, these are asked in your viva in lung viscera table also. So Clara cells are non-ciliated cells. Clara cells are present predominantly in terminal bronchioles. They replace the goblet cells in terminal bronchioles. They have many microvilli on the apical surface, and they secrete these two surface active protein and Clara cell secretory protein. Surface active agent reduces surface tension and reduces the collapsing tendency of airway passage. Clara cell protein is used as a marker for lung injury, and it is present in bronchoalveolar lavage fluid and serum. Clara cell provides protection against inhaled harmful chemicals, protect the alveoli, that is prevent emphysema, and work as stem cell also. Okay, stem cells mean it can give rise to other cells. Next is smooth muscle layer. It is predominant layer of the wall of the bronchioles. It is under control of autonomic nervous system and regulates the passage of air through the bronchioles. Connective tissue layer, bronchioles are surrounded by small amount of connective tissue and that separate the bronchioles from adjacent structures. Connective tissue is rich in elastic fibers. Okay. See, this is histology of lung. In the lung section, you can get sections of this bronchiole. Okay, intrapulmonary bronchus. This is section of intrapulmonary bronchus. These are sections of terminal bronchioles. This is section of respiratory bronchioles. Okay, respiratory bronchioles are directly connected to the alveoli, and these are the sections of alveoli. Alveoli are lined by flattened squamous cell, type 1 pneumocytes. Okay, and type 2 pneumocytes are few in number. They are helps in surfactant secretion. Okay, secretion of surfactants. They are cuboidal in shape. See here, respiratory bronchiole, alveolar duct. These are connected to the alveoli. And these two sections we have discussed, intrapulmonary bronchus and terminal bronchiole. This is a intrapulmonary bronchus longitudinal section. You can see some sections of cartilage is present, smooth muscle layer, lining epithelium. This is the lumen. Okay, and seromucous gland in the submucosa. So epithelium, lamina propria, muscular layer, submucosa, cartilage and adventitia. So all these 5 layers are present. It is intrapulmonary bronchus and discontinuous plates of hyaline cartilage is present. And this is terminal bronchiole, epithelium, lamina propria, muscular layer and this is the connective tissue layer, outermost. Devoid of cartilage plates, that is important point. Like this you will see under microscope in lung slide. Okay, these are the alveoli lined by simple squamous epithelium, pneumocytes. Okay, and this is alveolar duct leading to respiratory bronchiole. This is one section of the terminal bronchiole. Okay, epithelium, lamina propria, muscle, connective tissue. And these are all these are alveoli and respiratory part. Okay, so bronchioles have no cartilage, no seromucous glands. It has these 3 layers: mucosa, smooth muscle layer, connective tissue layer. Mucosa varies from larger bronchiole to respiratory bronchiole. In larger bronchiole, it is ciliated pseudostratified columnar epithelium with goblet cells. Medium bronchiole, simple ciliated columnar epithelium with goblet cells. Terminal bronchiole, there is simple cuboidal epithelium with Clara cells. Here goblet cells are replaced by Clara cells in terminal bronchiole. Okay, so location of Clara cell is terminal bronchiole. Understood? Respiratory, and you remember bronchiole means diameter is less than 1 millimeter. Okay, respiratory bronchiole is smaller than terminal bronchiole, less than 0.5 millimeter. Epithelium is simple cuboidal epithelium with Clara. So here one longitudinal section have been seen. These are the alveoli, alveolar duct, respiratory bronchiole lined by simple cuboidal epithelium with Clara cells, and this is terminal bronchiole, simple cuboidal epithelium with Clara cells. See, this is the cuboidal epithelium, rounded nuclei you can see, and outside in the lining of alveoli, flattened cells with flattened nuclei. Alveolar duct, alveoli lined by simple squamous epithelium, type 1 pneumocytes, type 2 pneumocytes and brush cells are seen in the lining of alveoli. Okay. So respiratory bronchiolar unit. The respiratory bronchiolar unit is the smallest functional unit of lung, consists of single respiratory bronchiole and alveoli that it supplies. Respiratory bronchiole, it usually measures less than 0.5 millimeter in diameter, participate in gaseous action, hence called the respiratory bronchiole. Layers are simple cuboidal epithelium, smooth muscle cells, a layer of delicate connective tissue layer outside. Alveolar ducts and alveolar sacs are present. Each respiratory bronchiole gives rise to 2 to 3 alveolar ducts. Each alveolar duct gives rise to the space called alveolar sac, and that is surrounded by clusters of alveoli. The surrounding alveoli open into the alveolar sac. Respiratory bronchiole lead to alveolar duct, lead to alveolar sac, and it goes to alveoli. Okay. Next we go to alveoli. Alveoli are thin-walled, polyhedral, sac-like terminal air spaces of the respiratory system that constitute the actual site for air exchange. Each lung contains about 150 to 250 million alveoli. Okay, this number is important for viva. Total surface area of alveoli is 75 meters square, equal to approximately surface area of a tennis court. Okay. Size of each alveolus is about 200 micron in diameter. There is alveolar septum in between the two alveoli, and interalveolar pores are known as pores of Kohn. Okay. Now, what are the cells of alveolar epithelium? This is asked in your viva. Okay. Cells of alveolar epithelium. The alveolar wall is lined by type 1 pneumocytes, type 2 pneumocytes, and occasional brass cells. Type 1 pneumocytes are present. 95% of cells are type 1 pneumocytes, flattened squamous cells. Type 2 pneumocytes are present. 5% cells are type 2 pneumocytes, and brass cells are occasionally present. These are cuboidal type 2 pneumocytes, and they help in secretion of surfactant. They produce surfactant, and this name of surfactant is important. DPPC, dipalmitoyl phosphatidyl choline. This is very frequently asked in viva and OSPE. Okay. Scientific name of the surfactant, dipalmitoyl phosphatidyl choline, also important for MCQs. Type 2 pneumocytes, 5% cases it is present. Function, it acts like progenitor of type 1 cells. That means it helps in generation of type 1 cells if they are get destroyed in due to some reason. Produce surfactant. Clara cells also produce surfactant. Okay. Brass cells, few brass cells are also present in alveolar epithelium, and these cells act as receptors to monitor the quality if the air entering in the alveoli. Okay, air quality is checked. So, different types of cells in the alveoli, cells in respiratory epithelium. In trachea, bronchus and alveoli, these cells are present and their function. Ciliated cells move the mucus, goblet cells produce mucus, basal cells give rise to other cells, granule cells, endocrine cells, brass cells are sensory, Clara cells, the air quality was checked by brass cells, Clara cells produce surfactant. And in alveoli, type 1 alveolar cells or type 1 pneumocytes, simple squamous cells helps in gaseous exchange. Type 2 alveolar cells, type 2 pneumocytes produce surfactant, brass cells are sensory cells. Okay, so these are the cells present in different areas of the respiratory tract. Okay, and tissue macrophages, they phagocytose the dust particles or RBCs. If they phagocytose RBCs, they are called heart failure cells, and if they phagocytose dust particle, then they are called dust cells. Okay. These are also present in some areas. See, the different type of cells has been shown. This is basal cells, smaller in height. Ciliated cells, cilia is present. Goblet cells, mucin material is present, which is released and appears empty loop. Brass cells, blunt microvilli are present. Clara cells producing surfactant. Granule cells, rough endoplasmic reticulum, granules are present, and type 1 pneumocyte is flattened cells, squamous cell, and type 2 pneumocytes, it also helps in surfactant secretion. Okay, understood? Alveolar macrophages are present in connective tissue as well as in alveolar lumen, and alveolar macrophages are the first line defense against lung infection. Main function of these cells is phagocytosis, and they perform this following action: removal of dust particles, hence called dust cells, removal of carbon and tar in smokers, removal of pollen grains, and removal of RBCs in heart failures. That's if they phagocytose RBC, it is known as heart failure cell. Okay, this comes in your viva and MCQ. What is heart failure cell? Okay, when the macrophages phagocytose RBC in respiratory epithelium, they form the heart failure cells. Okay. Next is blood air barrier. Okay, this is also important short note for how it is formed. The gaseous exchange between the alveolar air and blood occurs through the blood air barrier. The thinnest blood air barrier consists of this following component: thin layer of surfactant, simple squamous epithelium of lung alveoli. Third and fourth is fused basal lamina of alveolar epithelium and capillary endothelium, and continuous non-fenestrated capillary endothelium. All these are together forming the blood air barrier. Okay, if it comes as a short note, you have to draw a schematic diagram like this. You can see the layers. This is the surfactant, then there is flattened type 1 pneumocyte, then the basal lamina of the alveolus and the basal lamina of the capillary endothelium, and the squamous cell of lining epithelium of the endothelium. All these are together forming the blood air barrier. Okay, understood? Here you can see type 2 pneumocytes, cuboidal, okay, surfactant secretion, and these are the flattened cells are the type 1 pneumocytes. These are sections of blood vessels. You can see the RBCs within the blood vessels. This is another section of alveoli. This is one alveoli. This is another alveoli. Okay, understood? And here is the interstitium containing fibroblast, collagen fiber, elastic fiber, WBC, macrophage, like this. These are the difference between bronchus and bronchiole. Bronchus more than 1 mm diameter, bronchiole less than 1 mm in diameter. Epithelium pseudostratified ciliated columnar epithelium with goblet cells. Here larger bronchiole, simple columnar cells with few cilia and few goblet cells. Small bronchiole, simple columnar or simple cuboidal cells without cilia, without goblet cells. Smooth muscle layer present between mucosa and cartilage. Here present between mucosa and adventitia. Cartilages are irregular plates of cartilage present. Here cartilage is absent. Seromucous glands are present in submucosa. Here glands are absent. So these are difference between bronchus and bronchiole. It comes in theory or asked in your viva and OSPE. Okay. So these are clinical aspects. Pneumonia is lung infection, lung cancer, and metaplasia. Metaplasia occurs in cigarette smokers. Okay. Lining epithelium changes from pseudostratified ciliated columnar epithelium to stratified squamous epithelium, and it will lead to cancer. Acute respiratory distress syndrome, ARDS, or hyaline membrane disease. Bronchitis, infection of the bronchi. Immotile cilia syndrome, also known as Carter-Jenner syndrome, it occurs in some genetic conditions. Okay. And emphysema, there is dilatation of the alveoli and the respiratory part. Okay, so you have to draw a schematic diagram of lung in your histocopy and write down these 2 points. Presence of alveolar sacs and septa lined by simple squamous epithelium, presence of capillaries, terminal and respiratory bronchioles. All these are present in lung section. Okay, so you take a picture of this, you write, you draw a diagram, schematic diagram of lung in your histocopy. Lung and also a diagram of trachea, larger diagram. Here you can see intrapulmonary bronchus. These are sections of terminal bronchioles, sections of respiratory bronchioles, alveolar duct, and these are alveoli. Okay, understood? Division has been shown: trachea, principal bronchi, secondary, tertiary, terminal bronchiole, bronchiole, respiratory bronchiole, alveolar duct, alveoli. Tracheobronchial tree comes in your short note. You practice a schematic diagram like this. This we have discussed. See the pictures of intrapulmonary bronchus. Section of cartilage are present, lining epithelium, lamina propria, smooth muscle layer, some mucosa. You can see the glands and cartilage, and outermost is connective tissue. Okay, this is a high power view, okay, of the intrapulmonary bronchus. Epithelium, lamina propria, some mucosa, cartilage, connective tissue. These are the alveoli. This is one terminal bronchiole, lining epithelium. Okay, lining epithelium, here you can see simple columnar epithelium. Okay, no cilia, and lamina propria, muscle layer, connective tissue layer. These are the alveoli. This is section of vessel. Respiratory bronchiole, it is respiratory, there is connection with the alveoli. Okay. Lining epithelium is simple cuboidal epithelium, muscle layer and connective tissue. These are the section of alveoli. This is alveoli duct. It is also connected with the alveoli. No cartilage, no glands. This is alveoli. You can see the flattened cells. Okay. And here one type 2 pneumocytes, cuboidal cell is present. Here also another type 2 pneumocyte have been shown. Here is one dust cells have been shown. Macrophages have engulfed the dust particle to form the dust cells. Like this you will see under microscope: respiratory bronchiole, alveolar duct, terminal bronchiole, alveoli. So these are the cells of luminal epithelium of bronchioles: ciliated cell, basal cell, goblet cell, brush cell, small granule cell, Clara cell. Okay. This middle one is Clara cell. This is ciliated cell. Here is another ciliated cell. Enlarged view of bronchiole. Okay. Lining epithelium, lamina propria, muscle layer, connective tissue. So histology of trachea, you write down these 2 points and draw a schematic diagram like this. Identification points are mucous membrane lined by pseudostratified ciliated columnar epithelium. There is presence of hyaline cartilage and tracheal glands outside the mucous membrane. See, this is the cartilage, trachealis muscle, lining epithelium, some in lamina propria, then some mucosa containing the gland, seromucous gland. Here is the trachealis muscle. Outermost is connective tissue layer or adventitia, containing sections of blood vessels. Okay, and nerve. Okay, enlarged view of trachea, epithelium, pseudostratified ciliated columnar with goblet cells, lamina propria, some mucosa containing sections of seromucous glands, cartilage, you can see the cartilage cells, and perichondrium, adventitia. Okay, understood? Okay, like this you will see under the microscope. This is serous, this is mucous glands, okay, mucus acini, and this is serous acini. Serous acini are deeply stained, mucus are lightly stained, and these are excretory ducts, sections of blood vessels. Here this C-shaped cartilage section through the trachea, C-shaped cartilage. In this side trachealis muscle, posterior to trachea, this is section of esophagus. Okay. And on the 2 sides, these are the thyroid gland in front of the trachea, and there are vessels. This is common carotid vessels, vessels within the carotid sieve. This is low power view of pseudostratified ciliated columnar. Like this you will see here. We only see low power view 10x. Okay. Hyaline cartilage is prominent. That's how you identify the trachea. And these glands are sometimes seen connective tissue and outermost adventitia. This is medium 40x. Epithelium is clearly seen. This is cartilage, connective tissue with glands in this submucous part. See, high power view, the epithelium has been focused. Okay. So pseudostratified ciliated columnar, cilia you can see towards the lumen, and nuclei, these are the nuclei lying in different layers, all are reaching the basement membrane. And here is connective tissue. Like this you will see, this is epithelium, connective tissue. Here is the seromucous gland in submucosa, cartilage. You can see the cartilage cells are lying in groups, lined by the perichondrium having fibrous and cellular layer. Here is the adventitia outermost. Okay, so these are the layers you will see in trachea. And this is one section of respiratory bronchiole. Okay. So you see the sections, they are lining epithelium like this.
Terminal bronchiole.
It is this one is terminal bronchiole. This one is respiratory bronchiole. Okay, understood? Here on this side, in the upper part, you can see some sections of alveoli lined by flattened squamous epithelium. On this side, the rounded structure is sections of terminal bronchiole. Okay, you can see a layer of muscle in the wall, and here the longitudinal section of the respiratory bronchiole is present. You can see the alveoli duct is connected with the alveoli, and respiratory bronchiole is also connected with the alveoli. হ্যাঁ ওই লম্বা দেখে এটাই হচ্ছে রেসপিরেটরি ব্রঙ্কিউল, বুঝতে পেরেছিস? ওকে, দিস ইজ সেকশন অফ ট্রাকিয়া। ওকে, section of trachea you see the epithelium, pseudostratified ciliated columnar epithelium with goblet cells. Okay, this is a section of trachea. You see the epithelium, pseudostratified ciliated columnar epithelium. After that, lamina propria and some mucosa. In some mucosa, some glands can be seen. After that, you see the cartilage. Cartilage is very prominently seen here. You see the chondrocytes, matrix and the perichondrium. After that, outermost is the adventitia, loose connective tissue. Sections of blood vessels can be seen. Okay, okay. You only move the fine screw, no other thing you touch. White area is the lumen. Then there is the epithelium layer, thick layer. Okay, epithelium is pseudostratified ciliated columnar epithelium with goblet cells. After that, loose connective tissue layer. It is lamina propria and some mucosa. Okay, in the some mucosa, some glands you can see. After that, you see the cartilage layer. Cartilage is very prominent. You can see the chondrocytes lying in groups. Okay, and the matrix and lining perichondrium containing fibrous layer and cellular layer. Okay, and outermost is the adventitia, that is connective tissue layer. And within this adventitia, you can see sections of some blood vessels.